Imatinib is a tyrosine kinase inhibitor that acts on Philadelphia-positive BCR-ABL tyrosine kinases present in many forms of cancer, such as acute or chronic myeloid leukemia, acute lymphoblastic leukemia, and gastrointestinal stromal tumours. Its main use is in the treatment of CML by blocking the production of growth factors made by the BCR-ABL gene cascade, and is given as a starting dose of 400 mg once a day, which can be increased to 600 and 800 mg accordingly. Imatinib is a fairly polar planar molecule. It has 7 hydrogen bond acceptors, 2 hydrogen bond donors, and a log P of 3.47. The main way in which imatinib binds to the receptors is through hydrogen bonding interactions with threonine, methionine, aspartic acid, glutamic acid, and histidine. And finally through formation of a hydrophobic pocket with valine, leucine, and isoleucine. Imatinib exists as the salt imatinib mesylate as it increases the hydrophilicity of the drug and as an extension the solubility. Imatinib is used to treat leukemias with a Philadelphia-positive BCR-ABL gene. Leukemia is a type of cancer in which leukocytes in the bone marrow undergo uncontrolled proliferation and do not mature, causing them to be defective. This affects both myeloid and lymphoid cells, which specialise into other cell types such as erythrocytes, leukocytes, and platelets. The leukocytes can become cancerous through the BCR-ABL gene mutation. The BCR-ABL oncogene is formed through the translocation of the bronchocluster region and enables some proto-oncogene 1 gene sections. This can lead to the formation of the Philadelphia chromosome, which is present in 90% of patients with CML. The Philadelphia chromosome leads to the entire long arm of chromosome 22 being deleted, and chromosome 9 having an additional terminal band of approximately the same length. As a result of this, the first exon of the ABL1 gene is replaced by a sequence of the BCR gene, leading to the expression of the BCR-ABL fusion protein, encoding for a tyrosine kinase that is always on. This leads to constant activation of growth receptors and their downstream signal transduction pathways, causing uncontrollable cell division, proliferation, and damage to normal healthy cells and platelets produced in the bone marrow. These defective leukocytes will then not be able to fight off infection, and due to their destruction of other healthy cells, patients will find it hard to carry oxygen efficiently around their body or form blood clots. Imatinib is a type 2 tyrosine kinase inhibitor. These inhibitors can only bind to their targeted kinases, in this case the BCR-ABL kinase, when they are at a catalytically inactive or DFG out state. This is because in the inactive formation of BCR-ABL, the aspartic acid phenylalanine glycine site at the N-terminus of the activation loop, also known as the DFG motif, is flipped out. This exposes the allosteric binding site for imatinib. Imatinib is preferable to use over other anti-cancer drugs, as type 1 inhibitors directly bind to the ATP binding site, whereas type 2 inhibitors bind to the allosteric site adjacent to the ATP site, which also blocks the ATP site. This gives it the advantage of higher kinase selectivity and decreasing side effects. When imatinib is not present, the cancer proteins are phosphorylated, which will then go on to initiate cell division of leukemia cells. However, when imatinib is present, the cancer proteins cannot be phosphorylated and remain inactive, preventing proliferation of leukemia cells. This process does not affect healthy cells, as they do not possess the gene, and so the targeted therapy only kills cancerous cells, and it was one of the first therapies to work in this way. Toxicity has been widely associated with anti-cancer therapies. Evidence suggests the toxicological effect of imatinib to be mild and generally well tolerated by most patients, presenting in the first 2 years with on-target effects which are reversible upon discontinuation of the drug. Patients may commonly present with general gastrointestinal disturbances, including abdominal pain, appetite changes, and diarrhea. They may also experience general side effects associated with cytotoxic drugs. Skin rashes are commonly presented, with 66.7% of patients experiencing them, and there seems to be a causal relationship due directly to the endothelial growth factor inhibition. This is an on-target effect due to the inhibition of the BCR-ABL tyrosine kinase, affecting healthy skin development. This is also an oversimplification of the actual process, as the actual mechanism of action is still unknown. Edema is commonly seen in imatinib, and diuretics are prescribed, as well as a low sodium diet to help combat this. In rare cases, hypertension is seen, and this is postulated to be due to the suppression of the vascular endothelial growth factor receptor. Various studies have been carried out with regard to cardiac toxicity, but there is little evidence to substantiate that imatinib causes these problems. However, they are commonly found in imatinib derivatives such as sunitinib and sorafenib. Bone mineralization and hyperkalemia are side effects of imatinib, but the mechanism is still currently being investigated. They are, however, seen as off-target side effects, as imatinib inhibits other tyrosine kinases, which causes these issues. Finally, evidence suggests the common on-target side effects follow a similar pattern and therefore can act as biomarkers, which can be a useful aid to combat cancer.
In the mid-1980s, it was discovered that chronic myeloid leukemia was associated with the expression of BCR-ABL kinase, a specific oncogene product. Oncologist Brian Drucker, a researcher at Oregon Health and Science University, had been working on CML since the early 1990s. To try and find a suitable inhibitory compound of this oncogene product, he collaborated with members of the tyrosine kinase inhibitor program at Ciba-Geigy, now known as Novartis. The team started by developing routine biochemical assays, a process which took approximately 5 years. The original aim of the search was to find inhibitors of protein kinase C. The screening of synthetic compound libraries revealed that 2-phenylamminopyrimidine showed selectivity in blocking ABL and PDGF receptor kinases. This was therefore selected as the lead compound. A 3' pyridyl group is added to the lead compound at the 3' position of the pyrimidine. An amide group was added to the phenyl ring, which provided activity against tyrosine kinases. A flag methyl group was attached to the diaminophenol ring to abolish the undesirable PKC activity. Finally, an attachment of N-methyl piperazine moiety increased solubility and oral bioavailability. The resulting compound was imatinib, produced 8 years after starting the program. Imatinib inhibited ligand-activated platelet-derived growth factor receptors in vitro and in vivo studies respectively. It inhibited the activation of TEL platelet-derived growth factors and the autophosphorylation which causes the cancer. However, imatinib did not affect other cancer transduction pathways. This is the reason why imatinib is targeted specifically for CML. These studies concluded that imatinib had an effective therapeutic dose range of 0.1 to 1 micromoles. However, above 10 micromoles, 32D cell growth was not inhibited. The optimal treatment in mice allowed them to see days of no Ku812 tumour growth. 4 of the mice saw regrowths at days 48 to 60. Novartis began further research on imatinib because it had the highest selectivity for growth inhibition of BCR-ABL expressing cells. Scientists found that imatinib was also an ATP competitive inhibitor at the ABL kinase, which was one of the reasons why Novartis pursued with imatinib. This started in June of 1998 to determine the maximum tolerated dose of imatinib in humans with doses of around 300 milligrams. This dose saw the equivalent of 1 micromolar dose in the in vitro stage in the blood plasma of humans. Results from phase 1 and 2 meant that the FDA approved imatinib as treatment for CML in the advanced phase of CML after the failure of IFN therapy. Patients on imatinib saw greater chance of survival but also greater quality of life. Based on this, imatinib became approved as a first-line treatment of CML in both the United States and Europe by their respective regulatory bodies. Imatinib can be synthesized by using either batch or flow methods. Batch synthesis involves putting the raw materials into the system all at once, and then the product is all discharged at the end, which creates inconsistencies and byproducts. Therefore, it is no longer used for synthesis because of its time-consuming and costly nature, leading to constant supervision in managing these impurities. Flow synthesis is used to manufacture imatinib. This process involves raw materials constantly being charged and discharged from the system throughout the duration of the synthesis. It is a quicker and more efficient process, creating fewer intermediates and requiring less purification. During the reaction, 4-chloro-methyl-benzoyl chloride undergoes an amide formation reaction with 1-methyl-2-bromo-4-amino-benzene to give the compound shown. The chloride group is then substituted with the secondary amine group found in DMF. Finally, using a palladium catalyst, imatinib is formed through substitution of the bromo group for a di-aryl group. To summarise, imatinib mesylate was one of the first anti-cancer therapies to specifically target cancer cells and not affect healthy cells. This is due to it being a tyrosine kinase inhibitor that binds to the BCR-ABL fusion protein's allosteric site to stop overproliferation of growth hormones. It's made through batch synthesis and was developed by Brian Drucker in the 1980s, and due to it having a low toxicity and well-tolerated side effects, it is still used today in the treatment of the myeloid leukemias.